This video is about cells and organs of the immune system and hematopoiesis in particular. So, the many cells, organs, and tissues of the immune system are found throughout the body. They can be classified functionally into two main groups. The primary lymphoid organs provide appropriate microenvironments for the development and maturation of lymphocytes. The secondary lymphoid organs trap antigen, generally from nearby tissues or vascular spaces, and are sites where mature lymphocytes can interact effectively with the antigen. Blood vessels and lymphatic systems connect these organs, uniting them into a functional whole. So carried within the blood and lymph and populating the lymphoid organs are various white blood cells or leukocytes that participate in the immune response of the cells. Only the antigen specific lymphocytes possesses the attributes of diversity, specificity, memory, and non-self recognition. Antigen specific lymphocytes. Antigen specific lymphocytes possess the attributes of diversity, specificity, memory, and non-self recognition, the hallmarks of the adaptive immune response. Other leukocytes also play important roles, some as antigen presenting cells and others participating as effector cells. Effector cells in the elimination of antigen by phagocytosis or the secretion of immune effector molecules. Antigen presenting cells and others participating as effector cells in the elimination of antigens by phagocytosis or the secretion of immune effector molecule. Some leukocytes, especially T lymphocytes, secrete various protein molecules called cytokines. These molecules act as immunoregulatory hormones and play important roles in the coordination and regulation of immune responses. Here we describe the maturation of blood cells, the properties of the various immune system cells, and the functions of the lymphoid organs. So first let's learn about hematopoiesis. All blood cells arise from a type of cells called the hematopoietic stem cells or HSC. Stem cells are cells that can differentiate into other cell types. They are self-renewing, maintaining their population level by cell division. In humans, hematopoiesis, the formation and development of red and white blood cells, begins in the embryonic yolk sac during the first week of development. Yolk sac stem cells differentiate into primitive erythroid cells that contain embryonic hemoglobin. By the third month of gestation, hematopoietic stem cells have migrated from the yolk sac to the fetal liver. So, all blood cells arise from a type of cell called the hematopoietic stem cell or HSC. Stem cells are cells that can differentiate into other cell types. They are self-renewing, maintaining their population level by cell division. In humans, hematopoiesis, the formation and development of red and white blood cells, begins in the embryonic yolk sac during the first weeks of development. Yolk sac stem cells differentiate into primitive erythroid cells that contain embryonic hemoglobin. By the third month of gestation, hematopoietic stem cells have migrated from the yolk sac to the fetal liver and subsequently colonize the spleen. These two organs have major roles in hematopoiesis from the third to the seventh month of gestation. After that, the differentiation of HSCs in the bone marrow become the major factor in hematopoiesis, and by birth there is little or no hematopoiesis in the liver and spleen. The migration of hematopoiesis during development from a series of early sites to bone marrow is important. So when we try to learn about that, hematopoietic activity, 3 months yolk sac will be higher, then 1 to 7 months liver and spleen, fetal hemoglobin is there, and after birth long bones along with cranium, pelvis, sternum, ribs and vertebra adult. Such, then I mentioned about that. It is remarkable that every functionally specialized mature blood cell arises from an HSC. HSC in the sense hematopoietic stem cell. However, the study of hematopoietic stem cells is difficult for two reasons. They are scarce, normally fewer than 1 HSC per 5 into 10 raised to 4 cells in the bone marrow, and they are difficult to grow in vitro. As a result, the knowledge we have about how their proliferation and differentiation is regulated remains incomplete. So that's a portion open for research. By virtue of their capacity for self-renewal, hematopoietic stem cells are maintained at stable levels throughout adult life. However, when there is an increased demand for hematopoiesis, HSCs display an enormous proliferative capacity. This can be demonstrated in mice whose hematopoietic systems have been completely destroyed by a lethal dose of X-rays, 950 radons, rad. 1 rad represents the absorption by an irradiated target of an amount of radiation corresponding to 100 ERGs per gram of target. So such irradiated mice die within 10 days unless they are infused with normal bone marrow cells from a syngenic or genetically identical mouse. Although a normal mouse has 3 into 10 raised to 8 bone marrow cells, infusion of only 10 raised to 4 to 10 raised to 5 bone marrow cells from a donor is sufficient to completely restore the hematopoietic system, which demonstrates the enormous capacity of HSCs for self-renewal. So early in hematopoiesis, a multipotent stem cell differentiate along one of two pathways, giving rise to either a lymphoid progenitor cell or a myeloid progenitor cell. So progenitor cells have lost the capacity for self-renewal and are committed to a particular cell lineage. Lymphoid progenitor cells give rise to B, T and NK natural killer cells. Myeloid stem cells generate progenitors of red blood cells or erythrocytes. Many of the various white blood cells, neutrophils, eosinophils, basophils, monocytes, mast cells, dendritic cells, and platelet generating cells called megakaryocytes. In bone marrow, hematopoietic cells and their descendants grow, differentiate, and mature on a mesh like scaffold of stromal cells, which include fat cells, endothelial cells, fibroblasts, and macrophages. So, stromal cells influence the differentiation of hematopoietic stem cells by providing a hematopoietic inducing microenvironment or HIM consisting of a cellular matrix and factors that promote growth and differentiation. Hematopoietic inducing microenvironment. Many of these hematopoietic growth factors are soluble agents that arrive at their target cells by diffusion, whereas others are membrane bound molecules on the surface of stromal cells that require cell to cell contact between the responding cells and the stromal cells. So during hematopoiesis, erythrocytes and many different kinds of white blood cells descend from the few hematopoietic stem cells in a complicated process that involves many steps traversing hierarchy of precursor populations. Why has such a complex process evolved to generate blood cells? Over a lifetime, a person will produce about 10 raised to 16 blood cells. This requires a great many cell divisions. John Dick has pointed out that cell division is error prone and provides an opportunity for the genome to suffer mutations, some of which may cause cancer. He has suggested that to make this potentially catastrophic events less likely, the blood forming system is organized as an indigenous hierarchy in which most of the proliferation takes place within more differentiated precursors rather than in the hematopoietic stem cell population itself. These progressively differentiating precursors are not self-renewing, and the mature blood cells they form are either incapable of division or only divide under special circumstances. Consequently, the chance of generating cancer in HSCs and their immediate descendants is lowered, even it is not reduced to zero. So, might be cancers. Then hematopoiesis is regulated at the genetic level. The development of pluripotent hematopoietic stem cells into different cell types require the expression of different cells of lineage determining a lineage-specific genes at the appropriate times and in the correct order. The proteins specified by these genes are critical components of regulatory networks that direct the differentiation of the stem cell and its descendants. Much of what we know about the dependence of hematopoiesis on a particular gene comes from studies of mice in which a gene has been inactivated or knocked out by targeted disruption, which blocks the production of the protein that it encodes. If mice fail to produce red cells or particular white blood cells when a gene is knocked out, we conclude that the protein specified by the gene is necessary for development of those cells. So knockout technology is one of the most powerful tools available for determining the roles of particular genes in a broad range of processes, and it has made important contributions to the identification of many genes that regulate hematopoiesis. Although much remains to be learned, targeted disruption and other approaches have identified a number of transcription factors that play important roles in hematopoiesis. Some of these transcription factors affect many different hematopoietic lineages and others affect only a single lineage, such as the developmental pathway that leads to lymphocytes. One transcription factor that affects multiple lineages is GATA2, GATA2. A member of a family of transcription factors that recognize the tetranucleotide sequence GATA, a motif found in target genes. A functional GATA2 gene, which specifies this transcription factor, is essential for the development of the lymphoid, erythroid, and myeloid lineages. As might be expected, animals in which this gene is disrupted die during embryonic development. In contrast to GATA2, another transcription factor, Ikaros, IKAROS, is required only for the development of cells of the lymphoid lineage. Although Ikaros knockout mice do not produce significant numbers of B, T, and NK cells, their production of erythrocytes, granulocytes, and other cells of the myeloid lineage is unimpaired. So Ikaros knockout mice survive embryonic development, but they are severely compromised immunologically and die of infections at an early age. Yet another transcription factor, BMI1. We learnt about GATA1, I guess, wait a minute. 2, GATA2. Then Ikaros, along with another one called BMI1. BMI1. Okay, BMI1 is a transcriptional repressor that is a key determinant of the ability of HSCs to self-renew. And the gene for BMI1, which is highly expressed in HSCs of humans and mice, is knocked out. The BMI deficient mice die within 2 months of birth. The cause of death is the eventual failure of the bone marrow to generate red and white blood cells. The bone marrow failure was traced to lack of self-renewal by HSC. So we learned some genes and transcription factors which regulate hematopoiesis, like GATA1, BMI1, GATA2, BMI1 along with Ikaros. Ikaros is a transcription factor, and GATA2 is a transcription factor, and GATA2 gene is also there. Then Ikaros is a transcription factor. Another one, BMI1, is a transcriptional regulator. I hope the point is clear. Then hematopoietic hemostasis involves many factors. Hematopoiesis is a steady state process in which mature blood cells are produced at the same rate at which they are lost. The principal cause of blood cell loss is aging. The average erythrocyte has a lifespan of 120 days before it is phagocytosed and digested by macrophages in the spleen. The various white blood cells have lifespans ranging from a day for neutrophils to as long as 200 to 300, 20 to 30 years. 20 to 30 years for some T lymphocytes. To maintain steady state levels, the average human being must produce an estimated 3.7 into 10 raised to 11 white blood cells per day. This massive system is regulated by complex mechanisms that affect all of the individual cell types, and ultimately a number of cells in any hematopoietic lineage is set by a balance between this number of cells removed by cell death and the number that arise from division and differentiation. Any one or a combination of regulatory factors can affect rates of cell reproduction and differentiation. These factors can also determine whether a hematopoietic cell is induced to die. Then programmed cell death is an essential homeostatic mechanism. Programmed cell death, an induced and ordered process in which the cell actively participates in bringing about its own demise, is a critical factor in the homeostatic regulation of many types of cell population, including those of the hematopoietic system. Cells undergoing programmed cell death often exhibit distinctive morphologic changes, collectively referred to as apoptosis. These changes include a pronounced decrease in cell volume, modification of the cytoskeleton, which results in membrane blubbing, a condensation of the chromatin, and degradation of the DNA into fragments. Following these morphologic changes, an apoptotic cell sheds tiny membrane-bound apoptotic bodies containing intact organelles. So macrophages phagocytose apoptotic bodies and cells in the advanced stages of apoptosis. This ensures that their intracellular contents, including proteolytic and other lytic enzymes, cationic proteins, and oxidizing molecules, are not released into the surrounding tissue. As a consequence, apoptosis does not induce a local inflammatory response. Apoptosis differs markedly from necrosis, the changes associated with cell death arising from the injury. In necrosis, injured cells swell and burst, releasing their contents and probably triggering a damaged inflammatory response. And each of the leukocytes produced by hematopoiesis has a characteristic lifespan and then dies as by programmed cell death. In the adult human, for example, there are about 5 into 10 raised to 10 neutrophils in the circulation. These cells have a lifespan of only a day before programmed cell death is initiated. A stable number of these cells is maintained by constant neutrophil production. Programmed cell death also plays a role in maintaining proper number of hematopoietic progenitor cells for erythrocytes and various types of leukocytes. Beyond hematopoiesis, apoptosis is important in such by immunological processes as tolerance and killing of target cells by cytotoxic T cells or natural killer cells. Details of the mechanism underlying apoptosis are emerging. The expression of several genes and accompanies apoptosis in leukocytes and other cell types. Some of the proteins specified by these genes induce apoptosis, others are critical as apoptosis progresses, and still others inhibit apoptosis. For example, apoptosis can be induced by thymocytes by radiation, but only if the protein p53 is present. By cell death are induced by signals from Fas, a molecule present on the surface of many cells, and proteases known as caspase take part in a cascade of reactions that lead to apoptosis. On the other hand, members of the BCL2 B cell lymphoma 2 family of genes, BCL2 and BCLXL, encode protein products that inhibit apoptosis. And interestingly, the first member of this gene family to be discovered, BCL2, was found in studies that were concerned not with cell death but with the uncontrolled proliferation of B cells in a type of cancer known as B cell lymphoma. In the case, BCL2 gene was near the breakpoint of the chromosomal translocation in a human B cell lymphoma. So, before moving on to that, let's have an overview of hematopoiesis. So, you might have heard about hematopoietic stem cell, which is having the capability of self-renewing. Later, that will produce myeloid progenitor, which will lead to dendritic cell. Then, some other progenitors are also there, like granulocyte monocyte progenitor, which lead to the creation of neutrophil, monocyte, and macrophage. Eosinophil progenitor, which will create eosinophil. then basophil progenitor, which will eventually lead to the production of basophil, then megakaryocyte produce platelets, erythroid progenitor, which produces erythrocyte, then lymphoid progenitor is also there, which produce natural killer cell, and T cell progenitor is there, B cell progenitor is also there, which produce B cell, and T cell progenitor produce T H helper cell, TC cytotoxic T cell, then dendritic cell. Self-renewing hematopoietic stem cells give rise to lymphoid and myeloid progenitors. All lymphoid cells descend from lymphoid progenitor cells, and all cells of the myeloid lineage arise from myeloid progenitors. And when we try to learn about another picture, comparison of morphologic changes that occur in apoptosis and necrosis. Apoptosis, which results in the programmed cell death of hematopoietic cells, does not induce a local inflammatory response. In contrast, necrosis, the process which leads to death of injured cells, result in release of the cell's contents, which may induce a local inflammatory response. So in case of necrosis, chromatin clumping, swollen organelles, flocculent mitochondria, disintegration, release of intracellular contents, and inflammation. But in apoptosis, mild convolution, chromatin compaction and segregation, condensation of cytoplasm, nuclear fragmentation, blubbing will be there, along with apoptotic bodies are produced, which will eventually produce some sort of phagocytosis. Then apoptotic body is released from the phagocytic cell. BCL2 gene was near the break point of a chromosomal translocation in a human B cell lymphoma. The translocation moved the BCL2 gene into the immunoglobulin heavy chain locus, resulting in transcription of BCL2 along with the immunoglobulin gene, with consequent overproduction of the encoded BCL2 protein by the lymphoma cells. The resulting high levels of BCL2 are thought to help transform lymphoid cells into cancerous lymphoma cells by inhibiting the signals that would normally induce apoptotic cell death. And BCL2 levels have been found to play an important role in regulating the normal lifespan of various hematopoietic cell lineages, including lymphocytes. A normal adult has about 5 liters of blood with about 2000 lymphocytes per mm cube, for a total of about 10 raised to 11 circulating lymphocytes. During acute infection, the lymphocyte count increase fourfold or more, producing a circulating lymphocyte level of over 4 into 10 raised to 11. The immune system cannot sustain such a massive increase in cell numbers for an extended period, so the system must eliminate unneeded activated lymphocytes once the antigenic threat has passed. So activated lymphocytes have been found to express lower levels of BCL2 and therefore are more susceptible to the induction of apoptotic death rather than native lymphocytes or memory cells. However, if the hematopoietic stem cells can be enriched, so continue to be activated by antigen. Signals received during activation block the apoptotic signal. As antigen levels subside, levels of the signals that block apoptosis diminish and the lymphocytes begin to die by apoptosis. So then hematopoietic stem cells can be enriched. E. Weisman and colleagues developed a novel way of enriching the concentration of mouse hematopoietic stem cells, which normally constitute less than 0.05% of all bone marrow cells in mice. Their approach relied on the use of antibodies specific for molecules known as differentiation antigens, which are expressed only by particular cell types. They exposed bone marrow samples to antibodies that had been labeled with the fluorescent compounds and were specific for the differentiation antigens expressed on the surface of mature red and white blood cells, but not on stem cells. The labeled cells were then removed by flow cytometry with a fluorescence activated cell sorter. After each sorting, the remaining cells were assayed to determine the minimum number needed for restoration of hematopoiesis in a lethally X-irradiated mouse. As the pluripotent stem cells become relatively more numerous in the remaining population, fewer and fewer cells were needed to restore hematopoiesis in the system. So when we learn about that, we could browse images of light micrographs of a normal thymocyte developed T cell in the thymus along with apoptotic thymocytes, scanning electron micrograph of normal and apoptotic thymocytes. As the pluripotent stem cells become relatively more numerous in the remaining population, fewer and fewer cells were needed to restore hematopoiesis in the system. Removal of the hematopoietic cells by selecting for the presence of differentiation antigens allowed a 50 to 200-fold enrichment of pluripotent stem cells. To further enrich the pluripotent stem cells, the remaining cells were incubated with various antibodies raised against cells like to be in the early stages of hematopoiesis. One of these antibodies recognized a differentiation antigen called stem cell antigen 1. Treatment with this antibody aided capture of undifferentiated stem cells and yielded a preparation so enriched in pluripotent stem cells that an aggregate containing only 300 to 32,000 cells routinely restored hematopoiesis in the lethally X-irradiated mouse, whereas more than 10 raised to 4 non-enriched bone marrow cells were needed for restoration. So, to further enrich the pluripotent stem cells, the remaining cells were incubated with various antibodies raised against cells likely to be in the early stages of hematopoiesis. One of these antibodies recognized a differentiation antigen called stem cell antigen 1. Treatment with this antibody aided capture of undifferentiated stem cells and yielded a preparation so enriched in pluripotent stem cells that an aliquot containing only 30 to 100 cells routinely restored hematopoiesis in the lethally X-irradiated mouse, whereas more than 10 raised to 4 non-enriched bone marrow cells were needed for restoration. Using a refinement of this approach, H. Nakushi and his colleagues have revised procedures so effective that in 1 out of 5 lethally irradiated mice, a single hematopoietic cell can restore both myeloid and lymphoid lineages. It has been found that CD34, a marker found on about 1% of hematopoietic cells, although not unique to stem cells, is found on a small population of cells that includes stem cells. The administration of blood cell populations suitably enriched for CD34 plus cells plus indicate that the factor is present on the cell membrane can reconstitute a patient's entire hematopoietic system. Lethally irradiated mouse, 950 rats, 2 into 10 raised to 5 unenriched cells restore hematopoiesis, mouse lives. React with F1 antibodies to differentiation antigens. Number of cells injected into lethally irradiated mouse. Survival rate 1 into 10 raised to 3 partly enriched cells restore hematopoiesis, mouse lives. Then react with F1 antibodies against SCA1. Then 30 to 100 fully enriched cells restore hematopoiesis, mouse lives. Stem cell progenitor cells. Differentiated hematopoietic cells are removed by treatment with fluorescently labeled antibodies specific for membrane molecules expressed on differentiated lineages but absent from the undifferentiated stem cells and progenitor cells. Treatment of the resulting partly enriched preparation with antibodies specific for SCA1, an early differentiation antigen, removed most of the progenitor cells. M monocyte, B basophil, N neutrophil, EO for eosinophil, L for lymphocyte, E for erythrocyte. So enrichment of stem cell preparation is measured by their ability to restore hematopoiesis in lethally irradiated mice. Only animals in which hematopoiesis occurs survive. Progressive enlargement of stem cells is indicated by the decrease in the number of injected cells needed to restore hematopoiesis. A total enrichment of about 1000-fold is possible by this procedure.